Good morning students. Today we will discuss 2 marks and 3 mark questions in 12th Biobotany Principles and Process of Biotechnology 4th lesson. Students, in this chapter already we have seen 32 marks and 3 mark question. Now we start with the 32nd question. 32nd question, that is, which organism is called natural genetic engineer? Why? 32nd question is, which organism is called natural genetic engineer? Why? Page number 77. Answer is in your Botany book, page number 77. Organism that is naturally called genetic engineer is Agrobacterium tumefaciens. Agrobacterium tumefaciens is called natural genetic engineer. Why means it is given in page number 77. That is 4.2, 5.2 in the heading indirect or vector mediated gene transfer. In that paragraph, come to the 14th line. Agrobacterium tumefaciens is given in the 6th line. Underline that Agrobacterium tumefaciens, that bacteria. Then this bacteria, that is Agrobacterium tumefaciens, this bacterium has the natural ability to transfer, that is in the 12th or 14th line. This bacterium has the natural ability to transfer tDNA region of its plasmid into plant genome upon infection of cells at the wound sites. So it is also known as natural genetic engineer of plants. Close the bracket. Question number 33. Define indirect or vector mediated gene transfer. For this, answer is given in page number 77. Page number 77. Indirect or vector mediated gene transfer. That is in the topic, under the topic 4.5.2. Indirect or vector mediated gene transfer is gene transfer is mediated with the help of a plasmid vector is known as indirect or vector mediated gene transfer. Close the bracket. And the vector used is Agrobacterium tumefaciens. That is the example. Then 34th question, what is screening and what are the 3 methods of screening recombinant DNA? Page number 77, 78. First, screening. Define screening. After the introduction of, under the topic 4.6 screening of recombinant, below that, open the bracket, after the introduction of rDNA into a suitable host cell, it is essential to identify those cells which have received the rDNA molecule. This process is called screening. Close the bracket. This is define screening. What are the 3 methods of screening recombinant DNA? Are blue-white colony selection method, that is in page number 78, heading. You open the bracket and underline the heading, blue-white colony selection method, first one. Second one, antibiotic resistant marker, 4.6.2, antibiotic resistant markers. Third one, replica plating technique, 4.6.3, replica plating technique, that heading. The next question is, define electrophoresis. Page number, electrophoresis, it is given in page number 78. Students, write down the page number 78. Electrophoresis. Electrophoresis is a separating technique used to separate different biomolecules with positive and negative charges. This answer is given under 4.6.4 molecular techniques, isolation of genetic material and gel electrophoresis. Then next is, what are the 36th question. 36th question. What are the 2 efficient methods used in agriculture to detect and diagnose plant pathogen? This is very important question. Page number 79. It is given in page number 79 in the green colored box. ELISA, enzyme linked immunosorbent assay, and DNA probes, PROBES, DNA probes are the 2 efficient method to detect pathogen in agricultural diagnosis. Then next question, define blotting and what are the three types of blotting? Page number 79. Answer is in page number 79. Define blotting. Under the heading 4.6.5 nucleic acid hybridization blotting technique. Blotting techniques are widely used. In that paragraph, come to the fourth line. Blotting refers to the process of immobilization of sample nucleic acid or solid support sample nucleic acid. Blotting refers to the process of immobilization of sample nucleic acid or solid support. Close the bracket. Then next is, what are the three types of blotting? Below that paragraph, southern blotting, northern blotting, western blotting. Two headings you should write. The next is 38th question. What are the uses of blotting techniques? Uses of blotting techniques. Page number 79. It is given in page number 79. Blotting techniques. Uses of blotting techniques. Page number 79. Blotting under the heading 4.6.5. First point. Blotting techniques are widely used as analytical tools for the specific identification of desired DNA or RNA fragments from large number of molecules. Second point is below the definition, blotting. Below the definition, the blotted nucleic acid are then used as target in the hybridization experiment for their specific detection. These 2 points you should write for uses of blotting techniques. Then next 39th question, define target gene. Page number 80. Given in page number 80, target gene. Under the heading 4.6.6, bioassay of target gene effect. In that, target gene is target DNA, foreign DNA, passenger DNA, exogenous DNA, gene of interest or insert DNA that is to be either cloned or specifically mutated. Close the bracket. Then next question is transfection. Define transfection. Page number 81. In page number 81, it is given in green colored box. Transfection. Introduction of foreign nucleic acid into cells by non-viral method is called transfection. Then next question, define genome. Page number 81. In page number 81, under the heading 4.6.7, open the bracket, the whole complement of genes that determine all characters of an organism is called genome. Or you can write this answer, you can write. Then otherwise you can write, if it is easy, you refer page number 91, genome. Third vocabulary given, no? In that third one, genome. The entire complement of genetic material of an organism is called genome. Right? This also you can write. The next question is, define genome project. 42nd question, genome project. Page number 81. Page number 81, genome project. Genome project means, define genome project. Genome project that is given under the heading 4.6.7, genome sequencing and plant genome project. In that, come to fifth line. Genome project refers to a project in which the whole genome of a plant is analyzed using sequence analysis and sequence homology with other plants. Close the bracket. And genome project have been taken so far: Chlamydomonas, Arabidopsis thaliana, rice and maize plant. Any one of the example you can study, or for 1 mark you can study all the examples. Then next question is, RNA interference define. Define RNA interference. Page number 82. RNA interference is given in page number 82. Under the heading topic 4.6.10 RNA interference, in that you come to 20th line. RNA interference is a biological process in which RNA molecule inhibit gene expression or translation. Close the bracket. Then next question is, define BASTA. Name the gene and BASTA herbicide tolerant plant. Define BASTA. It is given in page number 83. BASTA is given in page number 83. Students, for this, BASTA means under the heading topic 4.7.2 herbicide tolerant BASTA. It is given there, no? In that one, third line, you come to third line. Phosphinothrycin is the name of BASTA. BASTA is otherwise called phosphinothrycin. That is, define BASTA means BASTA refers to a non-selective herbicide containing chemical compound phosphinothrycin. Close the bracket. This is definition for BASTA. And next continuation of the question is, name the gene and BASTA herbicide tolerant plant. BASTA herbicide tolerant plant is given in fifth line of that paragraph. Medicago sativa is a plant that is having BASTA, that is the BASTA tolerant plant, and the gene present in the tolerance genes deciding the tolerance ability of the plant is PPT gene. PPT gene was isolated from Medicago sativa, PPT gene. So next question, 45th question, what are the advantages of BT cotton? Page number 83. Direct answer is given in page number 83. Advantages, only 3 points. Cotton yield increase due to effective control of bollworm. Reduction in insecticide using cultivation of BT cotton. Potential reduction in the cost of cultivation. The next question is, 46th question, what are the disadvantages of BT cotton? Below that advantages, it is given 4 points. Disadvantages of BT cotton. That is, from cost of BT cotton is high. From that point to ending point affects, for this last point is affects pollinating insect and thus yield. Close the bracket. Then next is, define BT crops. Page number 83. Define BT crops. BT crop, it is given under the heading 4.7.3 insect resistant BT crops. In that, instead of BT cotton, you should write BT crop is a genetically modified organism or genetically modified pest resistant plant variety which produces an insecticide activity to bollworm. Example, cotton, BT cotton. Then next is, that is BT crops. The next is 48th question, how evolutionary relationship of plant is assessed? Page number 81. How evolutionary relationship of plant is assessed? Page number 81, answer is given. Evolutionary relationship of plant is assessed using DNA content. Underline the words I am telling. That is, under the heading in page number 81, under the heading 4.6.8 evolutionary pattern assessed using DNA. In that paragraph, come to the second line, DNA content, similarities and difference in the DNA sequence. Point number 1, DNA content. Second point, similarities and difference in the DNA sequence. Third point, relationship are indicated in cladogram. Fourth one, genetic distance between two taxa. And it also show antiquity or modernity of any taxon with respect to one another. Close the bracket. This is the how evolutionary relationship of plant is assessed. The next is 49th question. Name the transgenic mustard. Name the technology used to produce it, how it is produced. For this, answer is given in page number 84. Transgenic mustard. Mustard means kadug. Dara mustard. It is given page number 84. Dara mustard hybrid. Name of the transgenic mustard is Dara mustard hybrid. DMH. And name the technology used to produce it. The technology is given in the 8th line. I think it is 8th line. It was created by using Burners Foster technology for genetic modification. Then next is how it is produced. It is produced by adding genes from soil bacterium that make mustard. A self-pollinating plant, DMH11, contains 3 genes: bar gene, Pernès gene, mustard gene, sourced from soil bacterium. This bar gene had made plant resistant to herbicide named Basta. Then next is define PHB. Define PHB. Name the microorganism which produce PHAs. Answer is in page number 85. Define PHB. PHB means polyhydroxybutyrate. It's a synthetic polymer or non-degradable and pollute the soil when burned, and dioxin in the environment which cause cancer. Close the bracket. This is defined PHB. And this PHB, polyhydroxybutyrate and polyhydroxyalkanoates, that is PHAs, which have several medical applications such as drug delivery, scaffold and heart valves. Then there are some microorganisms. Next paragraph. Several microorganisms have been utilized to produce different types of PHAs, including gram-positive like Bacillus megatherium, Bacillus subtilis and Conibacterium, Glutamycin, a negative gram bacteria group like Pseudomonas species, and Alkaligenes eutrophus. That's all. Close the bracket. The next is define bioremediation, page number 85. Define bioremediation is given in page number 85. Bioremediation is defined as under the topic 4.7.11 bioremediation. It is defined as the use of microorganism or plant to manage environmental pollution. Point number 1. Second point, bioremediation process applied to the removal of oil, petrochemical residues, pesticide or heavy metals from soil or groundwater. The next question is define phytoremediation, page number 86. It is given in page number 86. Phytoremediation is the use of plants to bring about remediation of environment pollutant. If plants bring about remediation of environment pollution, then it is called phytoremediation. Then what is microremediation? Define microremediation. Is the use of fungi to bring about remediation of environmental pollution. The next is define bioventing. Bioventing is a process that increases the oxygen or airflow to accelerate the degradation of environmental pollution. The next is define bioleaching. It is given in page number 86. Bioleaching is the use of microorganisms in solution to recover metal pollutants from contaminated site. The next is define bioaugmentation. Addition of selected microbes to speed up degradation process is called bioaugmentation. Students, so far we have seen 56 2-mark and 3-mark questions in the lesson. In 4th lesson, process and principles of biotechnology. Rest of the question we will continue, right? Students, now we will see 57th question. Define southern blotting. Answer is given in page number 79. Come to page number 79. Southern blotting. It is given under the topic 4.6.5 nucleic acid hybridization. That southern blotting, types of blotting technique, it is given southern blotting. The transfer of DNA from agarose gel to nitrocellulose membrane is called southern blotting. Transfer of DNA, underline, this is the highlighted word, DNA from agarose gel to nitrocellulose membrane. The next is define northern blotting. That also in page number 79 below the southern blotting. The transfer of RNA to nitrocellulose membrane. Transfer of RNA to nitrocellulose membrane is called northern blotting. Define western blotting. Electrophoretic transfer of protein to nitrocellulose membrane is called western blotting. Next, 60th question and the last question of this chapter is what are the material used to grow microorganisms like spirulina? Page number 71. Answer is given in page number 71, SCP. Take the page 71, SCP. That is SCP. SCP under the heading single cell protein. In page number 71, come to the third paragraph. Third line, wastewater from potato processing plants, straw, molasses, animal manure, and even sewage are the material used to grow microorganisms like spirulina in SCP production. Student, thank you for watching this video and study well, prepare well for the examination. Write two or three times all this question and get ready for the examination. God bless you. Thank you.